Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test, that is MSc Entrance Exam question paper for the subject Life Science, conducted in the year 22nd March 2024. If you want still previous year question papers of Life Science being answered, you can go to my channel Omni Bio Entrance. Go to playlist, select CUET. In that you will find answers with answers explained. If you are new to my channel, please do like, share and subscribe. So coming to the first question. The portion of transmembrane proteins that cross the lipid bilayer are usually. So the transmembrane proteins that are crossing the lipid bilayer, they are usually of what structure they are having. So they are alpha helices. Answer is option 1. Then enzyme with their function we have to match. Cytochrome oxidase. Cytochrome oxidase is in electron transport system. Topoisomerases, they change the linking number. Cohesines, they are involved in cell cycle, and pCNA are involved in DNA replication. So the answer here is option 3, that is A1, B2, C4 and D3. Then next one. Which of the following enzyme is inhibited by citrate in Krebs cycle? So it is phosphofructokinase 2 that is inhibited by citrate. Answer is option 1. Why is the presence of proline destabilizes alpha helix? Proline residues introduce destabilizing kink in the alpha helix. That's correct. In proline, the nitrogen atom is a part of a rigid ring and the rotation about the NC bond is not possible. That is also correct. The nitrogen atom of a proline residue in a peptide linkage has no substituent hydrogen to participate in hydrogen bond with other residues. That is also correct. Proline residues work as helix linkers. That is not correct, because helix linkers refers to a segment that connects two alpha helices. But proline residues destabilizes the alpha helix. They do not link. So D is not correct. So A, B, C is the right answer. Answer is option 2. Then next one. Which of the following are components of the lipid raft in plasma membrane? So components of lipid raft include cholesterol and sphingolipids, whereas glycolipids and phospholipids are not. So answer is option 1, A and B. Then which of the following enzyme is defective in galactosemia, a fetal genetic disorder in infants? So in galactosemia, the enzyme that is deficient is galactose 1-phosphate uridyl transferase. Answer is option 4. Then you have to match vitamin with the disorder. Vitamin A causes night blindness. Vitamin D causes is a fat soluble vitamin, B3. Then vitamin C causes curve, C2. Vitamin K is a blood clotting, so D4. Answer is option 2, A1, B3, C2 and D4. Identify the correct sequence of the given intermediates formed during glycolysis. So you have to order the correct sequence. So lactate will come in the end. Phosphoenol pyruvate will come before lactate. So DC will be the ending. Now you have to see whether fructose 1,6-biphosphate comes or fructose 6-phosphate comes. So it is fructose 1,6-biphosphate, then fructose 6-phosphate. So BA. So BADC. So the answer is option 3. Identify the correct sequence of enzymes involved in TCA cycle. Here you have to know the TCA cycle. So first one will be aconitase, A. Then comes alpha ketoglutarate dehydrogenase, AD. Then fumarase and malate dehydrogenase. So ADBC. Answer is option 2. Which of the following is not a fluorescent dye? Silene silanol is not a fluorescent dye. Then the technique used to identify the specific DNA in bacterial colonies is. So it is bacterial colonies they have specifically mentioned. So it is colony hybridization. Then which metal is used for the electron beam generation in scanning electron microscopy or SEM? So it is tungsten that is used for electron beam generation in SEM. What is the role of sodium dodecyl sulfate in protein electrophoresis? Add net negative charge to the protein. Yes. Separate lipids, not lipids, proteins. So B is not correct. Modify native conformation of a protein. That is also correct. They disrupt hydrogen bonds, hydrophobic interactions and other forces. Then the protein lose their tertiary quaternary structure, adopting random coil or rod-like shape because of SDS. So C is correct. Allows direct destaining of the proteins. No, SDS page followed by staining allows for visualization of the protein band but not direct destaining. So A and C only are the right answers. Answer is option 2. Then next one, isoelectric focusing allows separation of the protein according to their PI, isoelectric points. Then technique with what they are used. You have to match. Western blotting, it is for protein identification, A2. Southern blotting is for DNA identification, B1. Northern blotting is for RNA identification, C4. Yeast 2 hybrid system is for protein-protein interaction studies. So answer is option 1, A2, B1, C4 and D3. Arrange the steps involved in polymerase chain reaction. Everybody knows this. It is denaturation, annealing, extension and then cycling. So answer here is option 1, A, B, C, D. Vertical gel electrophoresis is routinely used for the separation of proteins. Answer is option 1. Which is not a unit of radioactivity? Gray, Rontgen, millicurie, Pascal. Pascal is not a unit of radioactivity. It is a unit of pressure. In the technique called centrifugation, the centrifugal field is calculated as. So G is equal to omega square R. Answer is option 2. Then DNA or RNA modifying enzymes with their reactions you have to match. DNA is 1. It produces single-stranded nicks in DNA, A3. RNA is A. RNA is A cleaves and digests RNA and not DNA. S1 nuclease degrades single-stranded DNA and RNA. RNA is H. Cleaves and digests the RNA strand of RNA DNA heteroduplex. So A will be 3, B 1, C 4 and D 2. Answer is option 2. Number of chromosomes present in Arabidopsis thaliana is option 2, that is 10. What percentage of genes are found in the human genome? So it is 30% of the genome. Answer is option 2. Then next one. What are histone erasers? Are enzymes that remove specific histone modification, reversing chromatin remodeling and regulating gene expression. They are histone erasers. So they remove specific histone modifications. So A2, that is they are deacetylases. A2. Then what are histone readers? So histone readers are proteins that recognize and bind to specific histone modifications. So histone readers are proteins that recognize and bind to specific histone modifications. So bromodomain proteins, B4. What are histone writers? Histone writers are enzymes that add specific chemical modification to the histone. So they are methyl transferases. C1. C1 and DNA unwinding is by topoisomerase D3. So the answer here is option 1, A2, B4, C1 and D3. Then nucleosome is basically composed of. So nucleosome, it has got a histone core. H2, H2A, H2B, H3, H4A, H4B. If I remember, I have got the picture in one of the slides. I will show you there. So it has got a histone core. There is no linker histone in nucleosome. There is core DNA, but there is no linker DNA. Linker histone and linker DNA, they link two nucleosomes together. So A and C only. Answer is option 4. Then arrange according to their lower to higher genome size. So first one will be X 174, C. Then comes T7, then lambda and then T4. So the answer here is option 3, C, D, A, B. So with the addition of a linking number by +2 to a relaxed DNA consisting of LK is equal to 200. So initial relaxed DNA LK is 200. So twist is also 200. Rith is 0. It is a relaxed DNA. Then adding linking number by +2. So you get LK is equal to 202. The twist remains constant, but Rith increases by +2. So it increases positive supercoils. So the answer here is DNA undergoes positive supercoiling with LK202. Answer is option 1. What are the function of topoisomerases? So they change the linking number of a DNA. We already saw that in one of the match the following in the previous question. So topoisomerase 1, they cause a single strand DNA cut. Then relaxation of positive supercoils or introduction of negative supercoils. They will change the linking number thus. Then topoisomerase 2, they cut both DNA strands and change the linking number by plus or minus 2. So out of the options given here, repair chromosome ends, no. Form DNA isomers, no. Change the linking number of DNA is the right answer. Answer is option 3. Then next one, arrange the given process involved in nitrogen cycle. Ammonia formation, organic nitrogen formation, soil organic nitrogen formation, nitrate formation. Organic nitrogen formation first. Then comes soil organic nitrogen formation. Then ammonia formation and then nitrate formation. Answer is 4, B, C, A, D. Then next one. The complete proteins produced by an organism at any one time is called the proteome. Answer is 2. Retroviruses are enveloped viruses that can be engineered to deliver how many bases of single-stranded RNA? So they can deliver around 8,000 bases of single-stranded RNA. Then next one, adenoviruses are frequently used in gene delivery experiments. Their average genome size is 36,000 base pairs. Of their total DNA content, up to how much of adenovirus genome can be replaced by foreign DNA? So around 30 to 40% of the adenovirus genome can be replaced by foreign DNA. So 40% of 36,000 will give you 15,000 base pairs. That is my answer. But as per your answer key, it is 30,000 base pairs. So you go with 30,000 base pair. Answer is 4. Then next one. Gene gun is one of the popular physical delivery methods. Microparticles that are loaded to deliver DNA are made up of which metal? So in gene gun, it is made up of gold. Answer is option 1. Then next one. While delivering DNA by chemical method, one popular conjecture about what happens to polyethylenimine DNA complexes following endocytosis is known as the proton sponge hypothesis, which entails the following steps. PEI DNA complex are endocytosed. That is what they have mentioned in the question itself. Polyethylamine DNA complexes following endocytosis. So that will be the first one. PEI DNA complexes are endocytosed. Then what happens is V80 will pump proton into the vesicular interior. This will result in water enter into the vesicle to balance the osmotic gradient, and the vesicle will swell due to entry of ions and water leading to rupture. So the answer here is A, B, C, D, that is option 3.